everyone. My name is Maria Silveira. So this is your third stop, and this project was also funded through the Beef Cattle Enhancement Funds last year. And I want to introduce two people here that will help me with some demonstration. Yan Lu, she's a PhD student. She just joined my program last semester, and this will be the focus of her PhD dissertation. Amanda Baldo, she's a prospective master. She will help us with the demonstration how we collect water quality from these plots. So what you're looking here, we have 40 plots that were treated with either with biosolids or commercial fertilizer. So you see this control, the plot right next to it is a Class B St. Petersburg biosolids. You're not going to see much visual difference today because these plots were harvested a week ago. So you're not going to see much visual difference, but there's some difference in terms of the Bahia grass production. And the biochar material that we are using here is not biosolids. By the way, let me step back a little bit. What biosolid is? A lot of people call sewage sludge. So it's a human waste. But the term biosolids refer to the material that can be land applied. So sewage sludge is the raw material that comes out of the treatment plant. But this material that we are applying here, that we are referring as biosolids, it goes through several processes before it can be land applied. So there are two types of biosolids in Florida, produced and marketed in Florida, Class A and Class B biosolids. We have, in this study, we have a Class A pelletized material. It's a little bit easier to spread. It has much easier because, like I said, it's a dry material, it's a pelletized material. And we also have two Class B cake. One came from St. Petersburg facility, and one came from Bradenton. And we are applying this material, the biosolids and the commercial nitrogen fertilizer, along with this material that it's called biochar. So biochar is basically any plant biomass. This particular one is made from grass that goes through a pyrolysis process, which basically means a combustion in the absence of oxygen. And the end product, if you want to touch, you're welcome to. Your hands might get a little black because it's like an ash. It's ash. So one of the issues that we have, why we are evaluating co-application of this material with biosolids. Usually when we apply biosolids alone, biosolids contain a very high nitrogen to phosphorus ratio. So almost sometimes 1:1 or 1:2. When you look at the plant requirements, plants usually require 10 parts of nitrogen to 1 part of phosphorus, so a 1:10 relationship. So if I were to apply biosolids to supply my Bahia grass with nitrogen, what usually happens is you end up over-applying phosphorus, which from the plant standpoint, the plant won't cause any negative impact on the plant. But over time, you may end up in a situation where phosphorus tends to accumulate in the soil and can become an environmental issue. So the problem with biosolids is not because it's imbalanced or because it contains heavy metals or because it contains pathogen. The major issue that we often run is when people over-apply or they used this material improperly. So if it's used properly, it can replace commercial fertilizer. And like Chris Prevet was talking this morning about reducing cost, this is a very attractive option for producers because they can save a lot of money on fertilizer. And I don't know if you can still see, if you look behind you, there's a green spot over in that pasture. You can see the green area. That's where the biosolids that we used for this study were dumped. So you can see the difference when those pastures are fertilized with biosolids. It's an excellent source of nutrients. It contains nitrogen, phosphorus, micronutrients. It can also promote soil carbon accumulation in the soil, which can also can have a lot of positive impact on the plant because it helps hold water in the soil. It can hold nutrients in the soil. Like I said, you don't see much differences here because these plots were just harvested. So the goal of this study was to evaluate both the agronomic benefits, so we're looking at Bahia grass production, crude protein, and we're also looking at nutrient accumulation in the above ground tissue. A lot of people associate biosolids with heavy metals. In the past, biosolids contain a lot of heavy metals because the industrial waste was mixed with the urban waste. But that wasn't back in the 70s. After the Clean Water Act passed in the early 70s, urban and industrial waste are collected and treated separately. So we don't have trace metals or heavy metal issues in biosolids produced in the United States. When you go in countries like Brazil, yes, they still mix both sources, and then heavy metals can be an issue. In the United States, heavy metals are no longer an issue. The only difference between Class A and Class B is the amount of pathogens. So when you apply in Class B, there's some grazing restrictions. The animals cannot go back to the pasture immediately. There's a grazing restriction period. But in terms of the nutrition value, they are equally able to provide, especially nitrogen, to the plants. So we have a bunch of experiments that we're running here. For instance, one of the other limitations associated with biosolids use is that it's a limitation, and it's also an advantage. Not all the nitrogen present in the biosolid is immediately plant available. So the nitrogen needs to go through this process that's called mineralization before the plants can actually utilize. So the biosolids act like a slow release source of nitrogen. The only issue with that is if the producers don't apply enough biosolids to provide the nitrogen amount that the plants require, they might run into a deficiency issue. So one of the things that Yang is doing, she's evaluating how these different materials behave in terms of nitrogen mineralization rates. And I mentioned about this biochar. This biochar offers an opportunity to retain any phosphorus or even nitrogen that might escape the system. So because it's a carbon-rich material, I can pass this around. It's a carbon-rich material. It acts as a sorbent. So it retains any excess nutrients that might be in the soil. Of course, these plants, these perennial grasses, they can use much more nitrogen than what we are applying. For instance, in this case, we apply the biosolids at a rate of 160 pounds of nitrogen to the acre. We can apply twice as much this nitrogen rate that these plants will likely use all of that nitrogen. So in addition to the agronomic, I said the Bahia grass production, the nutritive value, and the nutrient concentration, we are also worried about the potential environment impact. So we are measuring two things here: water, the water cycle, and water quality. And we're also measuring gas emissions, nitrous oxide, carbon dioxide, and methane emissions. So we are comparing these different treatments. And what Yang and Amanda are going to do today, they're going to just quickly demonstrate how this is done. And those measurements occur every 2 weeks. So they come here, unless if there's a major, let's say a major rainfall event, Yang will come here right after the rainfall event, which, by the way, the past hurricane that was supposed to come here, she asked me, should I go there during the hurricane? I said, no. I know the data is going to be very interesting, but don't, safety first. Right now, Amanda's going to demonstrate I don't have we have very lysimeters that are installed at 3 feet deep. It's a large piece of equipment that took us, it was a lot of work involved installing these lysimeters. And they cost a lot of money too. They cost over $1,000 a piece. So that piece of PVC that's sticking out of the ground, those are our lysimeters. Amanda is going to demonstrate how to collect water samples. She won't be collecting water samples because right now the water table is below the depth that the lysimeter was installed. So now we're in drying season and we don't have any leaching going on. We don't have any vertical movement of water. But she's going to just mimic what she does when she comes to the field. And Ying is going to demonstrate the gas collection. If you want to come a little bit closer, please. The only thing you might get here is some biochar, but this is just ash. You guys need microphone? Let's open this. So what do we have for the gas collection? We have these anchors in the field because a lot of these gases are produced by microbes. So we don't want to physically disturb the soil when we collect the gas. So this is stay in the field. It's a little, probably 6 inches deep. When she comes to collect the gas sample, she puts this lid. Let me simulate. So basically, she's trying to seal this method is called static chamber technique. It's been validated, and a lot of scientists use the same technique to measure gas emissions from soil. So she usually puts a bunch of clamps here just to make sure that's airtight. She's not gonna do it today just for the sake of time. And then she has a timer here. So basically, the soil is emitting gas, right? And we put the lid, it's supposed to be airtight. There's a vent here just to make sure that the pressure doesn't build up inside the chamber. But then there's a port here where she collects the gas samples. And she collected over time, at time 0, and then she does, go ahead, 10 minutes, 20 and 40 minutes? 30. So just...
For example, now the first sample I will collect is 0. 0 means I collect 60 milliliter gas and then push into the little vial. The little vial, the volume is 10 milliliter.
So she put much more volume than the just to flush the air that was inside the vial.
Mm-hmm. And then this sample I will take to the lab to use the JC machine to analyze the three kind of greenhouse gas.
So for those who went to the lab, one of those fancy machines that I said it costs a lot of money, that's where we put this vial on, and the instrument runs the. Of course, it's not quite as simple as that, but anyway, that's how we analyze the gases present in this vial. So a lot of people say that about 1% of the nitrogen that we apply in the soil is emitted back to the atmosphere as nitrous oxide, and nitrous oxide is a very potent greenhouse gas. We want to see whether biosolids produce more or less relative to commercial fertilizer, or even plots that are untreated, that doesn't receive anything. My suspicion is we're not going to find any treatment effect, because again, these grasses can use so much nitrogen. The amount of nitrogen that we typically apply to these pastures, there's not much left to be lost as nitrous oxide. So the other thing we do is the water collection. So this is, the well is about, or the lysimeter is about 3 feet deep. Go ahead.
We
Go ahead.
just use the pump.
It's electric pump.
Put into the well, put into the bottom of the well, and then this part will connect a battery. And when we connect the battery, the water will come out. We use a container. We will record the volume of the water, and then I will collect this little bottle, 125 milliliter volume, and take to the lab to analysis. Use the AQ2 machine. I will analysis the OSP and nitrate and nitrate and nitrite, the concentration, and I will evaluate the water quality in each plot.
So the other thing I didn't mention, we have soil moisture sensors. They haven't been installed yet. They are in the lab. We can calculate whether this material, the biosolids and the biochar, can store. Oh yeah, she does that. This is another. I don't think we need to demonstrate. Every time that she collects the gas, she also measures the water, the soil moisture, and the temperature, because the gas emissions are directly related to those 2 parameters. But anyway, we're trying to estimate how much phosphorus and nitrogen is lost either in the water or as gas. Oh, there's some water in there. So, but it's also important to measure how much water is moving down in the soil profile, because that way we can also calculate the water budget. So by adding materials that contain high carbon, there's a potential to increase the water retention capacity of our soils. So in terms of production, when you think in the fall or in the times of the dry times of the year, this material can provide another benefit. Not only the nutrition that they are providing, the nitrogen and phosphorus, but they can also help these crops to thrive better during those stress periods. If you are interested, I know you probably want to move on to the next topic, but the girls dug a soil pit over there. It's a very nice, interesting, I think it's very interesting to see the soil profile. And they dug the pit right beside. You can see the hole here, and these are the different layers. So our lysimeter is installed somewhere in this layer here, about 3 feet. Usually when you look at this is the surface layer. This is where we usually do our soil testing. The phosphorus concentration in the surface layer is less than 10 parts per million. Very low phosphorus concentration. When you move down in the soil profile, and you can see the amount of roots that actually, you can see the roots that occur in these deeper layers, phosphorus concentration increases tremendously. So we go from less than 10 to something like 50, sometimes 100. So there's a lot of phosphorus. This is natural, just a natural process that the soils were formed in Florida. Phosphorus tends to accumulate in the deeper layers. So this is another important aspect. We spent this first whole year, this project, we didn't apply these amendments until probably August, because we want to generate a lot of background data. So before anything is implemented, before you apply any type of fertilizer, what is the phosphorus, how much of phosphorus and nitrogen is being leaching in the system, how much is being emitted to the atmosphere. Now that we're confident that we have a very good baseline data, we can see the treatment effect. But I want you to see how to me, it's very cool transition from one horizon to the other. And the water table, of course, for a good part of the summer, there's another piece of instrument that we monitor the water table I think it's recording every 15 minutes, so I can tell whether or not we should come here and collect water samples. But anyway, for a significant part of our summer, our water table was above the 3 feet mark. So we couldn't install some of these instruments. We have to wait for the water table to decrease before we could do some of this instrumentation. Any questions? So again, this is the type of topic that's very important to the producers here. But when you go to there has been a lot of research that's been conducted on biosolids, but they primarily been done in very controlled conditions, laboratory or greenhouse. There hasn't been any field scale study that address agronomic and environmental issues related to land application of biosolids. And by the way, when you drove by, that is not a research site, it's a demonstration site. These two pastures, they have 15 acres each. We have biosolids application versus ammonium nitrate. Every year we've been doing that. But we are not collecting very intensive data like we are doing here. Because for us to be able to do this very intensive monitoring, it needs to be a small scale. So in those large pastures, we have, for demonstration purpose, the two biosolids and the commercial fertilizer side to side. Any questions? I'm sorry, I said any questions and I started talking again.
What is the current withdrawal period for Pest B? How long do you have to stay off?
The question was how long was the grazing restriction? 60 days. I don't know if that varies from location to location, but DEP, we actually had to get a, not a formal permit, but I had to get the DEP blessing to be able to do this study and for the hauler to help me to bring the material here. So they told me 60 days, but this is not going to be grazed. We're mechanically harvesting every 6 weeks.
And there is none for A?
None for A. If the A is marked and labeled as fertilizer, there's no restriction.
And is it also it can come within a certain distance of water bodies and that sort of thing?
There is a restriction as far as the Class B, yes. I don't know off the top of my head what the distance is from water body. But there's some, yeah, they were concerned about a wetland, but it's kind of far from here. The DEP, they came here to visit the site to make sure that everything was in compliance. I was surprised because this is such a small area, and I was talking about 15-acre pasture, but it was such a big deal to get their blessing.
What was the rate on the biosolids?
The question was what was the rate of the biosolids? Like I said, not all the total. It was based on the nitrogen concentration, and we're targeting 160 pounds of plant available. But that doesn't mean that we apply. So we measure total nitrogen in the biosolids, let's say 5%. And DEP allows you to multiply your desired rate by a factor of 1.5, just to account for the mineralization factor. So we multiply the 160 by 1.5. So we end up applying biosolids at a rate of 240 pounds of total nitrogen per acre, which, assuming that 1.5 factor, that means we're going to have 160 pounds of plant available nitrogen for the first year. And in fact, the 1.5, of course, it varies from material to material. And this is another aspect that Yin is doing. She's going to incubate the samples, the different samples, in what we call liter bags, and she's going to put in the field and collect the samples periodically over time. to determine that mineralization rate. It could be, of course, a material that's being heat dried. Probably that we expect the mineralization rate to be much lower. So in other words, if the mineralization is lower, that means we can apply much greater rates. For materials that mineralize faster, maybe 1.5 can be cut down a little bit. So this is another aspect of her research. And there's a lot of interest. I received a couple calls from the regulatory agencies on the biochar aspect because it can sorb, it can retain some of the excess nutrients. They are also interested in that as being a potential management alternative. Of course, we co-applied both on the surface, and it was a lot of work. I don't think producers, from the practical standpoint, producers will not like to do that. But if maybe the municipality can add that amendment to the biosolids before it's land applied, might be an option, and maybe reduce the nutrient availability in those materials that have that nitrogen to phosphorus ratio too high. Yes, Eric.
Is biochar a byproduct of some process? I've not heard of it before.
It is a byproduct. So the one that we land apply, it was made from wood chips. And the physical characteristics was a little bit different than the material that we are passing around. But sometimes you can find for free. This particular one we pay, and it was quite expensive. But it has they're different materials. It could be grass, and sometimes some of the companies that are marketing, they can custom made for you. So I want some that were made from grass and some that were made from woody species, just to see, because it also, depending on the feedstock and the process, how these biochars were generated, the properties are also different. So Amanda is doing a lab work testing different materials. But yeah, it sounds like a very attractive option. The only thing is, because they can sorb, they can also compete with the plants for those nutrients. So the rate, a proper rate, and to what extent they're effective reducing nitrogen, those are the questions that we don't know. So we're trying to address this in the field study, and we have some additional laboratory study.
And you're using that to limit the phosphorus?
Not to reduce the phosphorus, but to reduce the availability of the phosphorus that's present in the soil. Because I envision that we're going to be doing this for many years. So eventually, I think we're going to end up building up phosphorus in the soil. So maybe that material can help. It's not going to minimize the load. The phosphorus load will still be there, but maybe it will minimize the availability. If this phosphorus is tied up with this highly carbon-rich material, and it's not moving, it's not leaching, it's not producing, not letting nitrogen to be lost to the atmosphere, so that might be an option. That's it.
Maria, how much of that Class B how many tons did you put down per acre? And what's the approximate cost of it?
It was a lot because the moisture was very high. It was almost 85% moisture, so it was it was a cake, but do you guys remember 24? For each plot that you can see here was about 30 pounds of the. I have to make the calculation.
I wonder what the cost is per acre for what you put out. You know, looking at it from a commercial standpoint.
It varies. I don't know off the top of my head because some material contain 1% nitrogen, some contain 5%. So the actual amount of biosolids, and because of the moisture content, so the 240 was on dry matter basis. So depending on the moisture and the nitrogen concentration, we end up putting different amounts. So that was one of the issues. They have to dump the material here. We have to collect a subsample, run in the lab. But this step needs to be done quickly because we didn't want the material to stay here for very long.
Would you know?
Yeah.
Would you know, is your St. Pete material the same as your mulberry material?
According to Cindy, they are not the same. I'm not. And let me tell you, there's a lot of misconception about biosolids, even among some of the staff here. When I said I was going to work with biosolids, everybody was scared. Oh, she's going to bring the stinky, nasty. But we have to remember, people don't seem to have problem with cattle manure or animal manure. But when it comes to human manure, there's a whole different deal. These materials, they have been processed and they meet the requirement to be land applied. So it's just a source of fertilizer, and in fact, it's a cheap source of fertilizer. So if we can take advantage and apply them in a way that doesn't cause any environmental problem, I think it's a win-win situation. It's much better environmental use to recycle in the pasture than dump in a landfill.